What is a stem cell? Well, real briefly, I think if stem cells had been discovered today, we would call them an unprogrammed cell. But since they were discovered over 30 years ago, there weren't that much computer programming back then. So the people who discovered them thought the best analogy would be a tree, and specifically the stem of a tree that was dividing. These scientific terms for these types of cells are called pluripotent or multipotent, and in the case of embryonic stem cells, you can also use the term totipotent. The easiest cartoon that I can show you that illustrates what a stem cell is a tree. And the stem cell is the trunk. And then the branches and the leaves at the end are the cells that now have lost their totipotent or multipotent potential, have lost their expressions of a stem cell, and now have the expressions of a differentiated cell. So in brief, stem cells are the tree trunk of the tree of life when it comes to cells. Now, how do you identify a stem cell? Because they don't just jump out and tell you that's what they are. There's basically two things you need. You need a microscope that has a fluorescent light, and you need fluorescent antibodies. Now, how many of you have heard the term CD before? Have you ever wondered what that stood for? I did too. It stands for clusters of differentiation. And basically, if you go back and look at the nomenclature, cells have been found to have markers on their surface. In fact, there's over 300 clusters of differentiation now that have been found. And most of these have nothing to do with stem cells. But there's one that seems to be ubiquitous on stem cells, and it happens to be number 34. So if you see something that says CD34+, that's usually a stem cell. Here's a CD34+ stem cell from fat. And once you have a microscope and you have those antibodies, you can now look at those cells and identify them. Here on your right are some stem cells that have been labeled with a fluorescent antibody that then fluoresces in yellow. And on your left is the graph from a cell counter that uses fluorescent markers to count cells. So once you have the antibody attached to these cells, it's a very useful way of identifying them and then quantifying them. Now, why all the fuss about stem cells? Well, I want to contrast your normal cells with stem cells, and I think you'll quickly realize why everybody is so excited about this. Normal cells cannot self-renew. Stem cells can.
It's about 3/4.
Normal cells cannot repair tissue. Stem cells can. Normal cells grow old, but stem cells do not. Even in older people, even as you age, your stem cells do not age. Normal cells cannot differentiate, except, of course, the stem cells in your body, whereas stem cells can differentiate. For that reason, I personally believe that stem cells have the most promise for treating disease, degenerative conditions, and aging of anything that has been discovered today. Now, for those of you who are a little older, how many times have you heard hyperbole like that? You remember when gene therapy came along? It was going to be the latest, greatest thing that was going to cure everything. Remember 10 years ago, the human genome sequence was sequenced, and we were all convinced that we were going to find the cure for cancer and aging and heart disease. And now 10 years have gone by, and we still don't really have a lot to show from sequencing the human genome. Well, I personally believe that this is going to be different, but I think the jury's still out on that. Now, here is the key to stem cells, is how are they transformed? The scientific word for transforming is differentiation. And basically what you do is you add inductive factors, depending on what you want these cells to deform. So this table here is a list of the inductive factors that have been shown to induce a stem cell to form a differentiated cell. I'm sorry, I apologize, the blue did not show up real well, but real briefly for those of you who can't see the blue. The first one there is muscle, next one is bone, cartilage, heart muscle, nerve, tissue, blood vessels, and fat. Basically, if you add these inductive factors, stem cells will automatically differentiate into those areas. Here's an example of some embryonic stem cells that 4 inductive factors were added to, And as you can see, they gradually transformed from a cell that had CD34 plus markers, and you can see the blue fluorescence, to a differentiated cardiac muscle cell where they no longer have the CD34 plus marker. And this all happened because of the inductive factors that were put in that Petri dish. To me, this is the most amazing thing, that these cells can transform like this and that we can actually influence them with these inductive factors.